This is a production of Cornell University.
So I'm here to talk to everybody about inoculum, Phytophthora infestans inoculum, which is a pretty basic part of all the experiments, but I'm going to make the argument that it's a pretty important part. And so yeah, the title is Does Inoculum History and Preparation Affect Pathogenicity? But to explain it a little bit clearer, when we're talking about Phytophthora infestans, when we're investigating Phytophthora infestans, does the substrate that we grow and harvest the sporangia that we use for inoculum, does that substrate affect the disease that's caused downstream in the experiments? Why? Does it really affect the disease phenotype that we get from this? And so the reason this is an issue, the reason that I thought that this would be worth investigating, is that among the community, among those of us that study Phytophthora infestans, there's not a lot of standardization. The way in which it's grown, the substrate that it's grown, is pretty variable. You can have it grown on live tissue, generally on detached leaflets, which is how we like to do it in the Fry Lab. It can be grown in pure culture on a whole bunch of different agar media. And then resulting from this variation, you often get these really disparate concentrations used for inoculum, for spraying plants or inoculating individual pieces of tissue, all the way from 300 sporangia per mil to upwards of 150,000 sporangia per mil, which is a lot. And this comes up. So this is a fairly recent paper out of our lab. And Paula in this paper was using inoculum derived from leaflets, from detached leaflets, how we generally do it, in our lab. And she said that she wanted to use it coming from leaflets because it would more closely represent the inoculum that we would see in the field than an actual infection coming from other plants. That's what we see, right? It doesn't come from cultures in the field. And that's generally the rationale used when we're trying to decide if we're going to use it from leaflets. And she used it at about 4,000 sporangia per mil, which is what we've been using things at in the lab recently. But this is, while this makes a lot of sense to me, and this is how I've done all my experiments, this certainly isn't it's not the standard, it's not the rule. Up here, this is actually the most recently published paper on Phytophthora infestans on the internet, at least, right? This came out last week. They got their inoculum from rye agriculturists, from pure culture. And they washed the sporangia from the culture, and they actually used concentrations very similar to what we use, about 4,000 sporangia per mil. But if you look at another paper, this is from 2008, these folks, again, washed their sporangia from pure cultures, from agricultures, but they inoculated their plants at 150,000 sporangia per mil, which is, that sporangia suspension in distilled water, it looks like milk. It's cloudy with sporangia. It's an astronomical amount of inoculum. It's crazy. You often get concentrations in the 50 to 60,000 range. These guys washed it from leaf tissue, from actual sporulating tissue. That's generally, if you wash a moist chamber's worth of tissue, this is the concentration you get in a falcon tube. It makes sense. But all the way at the low end of the spectrum, this is a much older paper, they were using 300 sporangia per mil to inoculate plants in high tunnels, actually. So there's this huge variation just to try and get disease. And so that in itself should say something that there's, this can't not be causing anything, right? When you've got, a difference of 1,000 times in the amount of concentration. And so we have other evidence to actually back up that the way that we get our inoculum, the substrate that it came off of, has a big impact on things. So this is some data from a recent study, some recent work that was done on arachidonic acid conferring a resistance phenotype to potatoes, right? So if you treat some potatoes with arachidonic acid and you have untreated controls, and then you apply Phytophthora infestans inoculum to them, right? You get a really notable difference in disease expression, right? So for this experiment, it was taken from pure culture. It was washed from rye agar, and they inoculated and assessed disease. And if you did this experiment and you got these results, and it was repeated in a well set up study, you would probably make the assumption, and I think you would rightfully make the assumption that, yeah, wow, we have a phenotype. This really, really does something. But if you repeat the same experiment down to the T, the only difference being that our inoculum of the same isolate now has come from sporulating plant material instead of coming from a pure culture. That phenotype we saw before is essentially gone. You could not draw the same conclusions from this experiment as you could from the one before. And the only thing that's different here is how we got our sporangia, right? And So I think that it's a really important thing to be considered, and it can often be overlooked in these experiments. And so really my hypothesis that I'm looking at for this whole thing is that the substrate that Phytophthora infestans is grown on, and that this sporangia is collected from, really does induce some kind of significant change to the sporangia, some sort of epigenetic change to the sporangia. And then that change then affects the disease downstream, the disease that we get in the experiments we use this inoculum for. And so how do we look into this? At its most simple, we compare the inoculum of some isolated Phytophthora infestans grown in pure culture and grown on live tissue, on leaf tissue, and do an inoculation experiment with it and see how the disease progresses. Not particularly complicated, right? And so the outline of the project, the way that it followed, is really kind of the way that we planned it and executed it. And so really the first goal is to do an inoculation experiment and actually confirm that there really is a phenotypic difference between inoculum that we source from actual plant material and inoculum that we source from pure culture. And that's just done by assessing the overall disease at a set time, right? And then I really wanted to look into the mechanisms behind this, try and look at the genetic differences that's going on here, to try and maybe see why we're seeing this difference. And so the big component of that was total gene expression in tomato tissue, right? When I do these infection studies, I can sample tissue on the time course and then extract the RNA out of that tissue, and that tissue has post-gene expression, right, tomato gene expression, and Phytophthora gene expression within it. And so I can look at the interaction that's going on. But then actually, as the experiment came together, and we did more thinking about it, it became apparent that actually the gene expression within that inoculum itself, the stuff that I would be spraying on the plants, is really important too. Just the pure concentrated inoculum coming from pure culture and coming from sporulating live tissue. And so looking into the gene expression of that inoculum became a really important part of the experiment. So the first part is actually assessing these inoculum sources, right? Actually looking at the difference. And so the considerations that I wanted to really keep in mind while I was planning the experiment was I wanted to use more than one isolate of Phytophthora infestans, and I didn't want to just be a fluke, just some artifact that only one isolate was representing. But those isolates had to all be within the same clonal lineage. Clonal lineages of Phytophthora infestans are pretty diverse. They have different infection rates. They have different preferences. They behave differently, so they had to be within the same clonal lineage. I wanted to keep it consistent, so I wanted all my material to come from Phytophthora that was collected in New York State on tomatoes. I was going to do the experiment in tomatoes, and so I just wanted to keep that consistent. And then I really wanted to keep an eye on inoculum concentration. We saw that there's a huge variation in inoculum concentration. And change in concentration really can impact the way that the disease goes forward. So I wanted to make sure that it was pretty consistent. So the two isolates that I ended up using for this experiment were both US-23s. The first one, which you can see on the top here, I referred to as Newark Valley, and I referred to as Newark Valley for the rest of the experiment, simply because it was collected from Newark Valley. It came in as a disease sample to be checked for clonal lineage to be identified by our lab. It was sent to Kev. It came in just this past summer, last June, truly less than weeks before I used it in the experiment. It was really, really fresh from the field. And to support this experiment, I selected an isolate called 140112. That's just our lab nomenclature. It's how we keep track of our isolates. It was also a US-23 collected from tomato in New York State, almost exactly a year before we got Newark Valley. So it had been kept the way that we keep all our cultures in the lab, on rye agar transferred periodically. So with my two starting cultures, 140112, which is in its plate, its storage medium, and Newark Valley, and off of a field sample, they had to be transferred, to the differential media, right? So 140112 I needed growing on in pure culture and on leaflets, and Newark Valley I needed growing in pure culture and on leaflets. But this first step here is actually no trivial task, as anybody who's worked with Phytophthora can attest to, and as Bill said, something that I struggled with as an undergraduate before I came here significantly. Primarily this step and this step, so getting storage Phytophthora off of a slow-growing media onto leaflets is not it's not as simple as just putting an ice cube on it and letting it go. It actually is a bit bit of work to get it going. And especially, you know, we've only got maybe 1 or 2 plates in storage. We don't have a lot of plates to make a bunch of inoculant to spray leaves. And this is a non-trivial task too, getting it actually from a field isolate into culture. If you just, you know, bring a piece of tissue over, you end up with contamination and all sorts of horrible things. We have to use, you know, agar cubes and actually transfer sporangia. And I really have to give all the thanks for this to Kev. Kev processed all these samples, and he really got everything going, which was wonderful. So after I get these going, this can be thought of as like a lab stock. I don't want to use that as the first source of inoculant because it's just been transferred, these agar cubes and stuff in the way. So then these are transferred out to their equivalent media one time. And then this is the inoculant source for the first field trials. So I had field trials and lab trials, because when we look at Phytophthora infection experiments, there are field trials, right, where people spray plants in a farm setting. And there are lots and lots of lab experiments with individual leaflets. And so I had roughly 5 trials going on, and I needed a lot of inoculum. So the inoculum was prepared on their respective plates and leaflets, and then flooded from their source and used as inoculum in the field. And it was also used to inoculate the next generation of inoculum in the next trial. So after it was washed, I adjusted the concentration to 5,000 sporangia per mil, which is pretty similar to what other experiments we were using in the lab was. It was the same concentrations I was using for other experiments I was doing at the time. The sporangia are incubated in the fridge for half an hour to promote germination and zoospore release, because that really helps with getting infection later down the road. And then after they had germinated, I put them back on the hemocytometer under the microscope to double check concentration. I really wanted to make sure the concentrations were all the same. But also to assess germination, I wanted to make sure that, you know, I wasn't seeing that the leaflet-derived sporangia were all germinating happily and lots of zoospores were being released, and the plate ones had just not germinated. That in itself could be causing a difference. And luckily, actually, there wasn't much of a difference. They were all happily germinating. It was really nice. So then those suspensions were put into an aerosol sprayer, just like this one, and brought out to the field or into the lab to inoculate my plants. So I used 5 mils total of that concentration in the field plots because they were multi-plant plots that were big, and only a mil for the lab experiments because they were just plates, right? They were much smaller. So it was the same amount of material basically per leaf. I had calculated that way. So for the experiments, I had 2 field trials that were done at the farm down in Freeville this past summer. The 2 trials were comprised of 3 blocks each, randomized complete blocks, and there was essentially 10 treatments. We can think of it as 5 treatments doubled. And those treatments were a water control, right, to make sure that, you know, I had a healthy plant reference. 140-112, the older isolate from leaflets, 140-112 from pure culture. Newark Valley, the newer isolate from leaflets, and Newark Valley from pure culture. So those were 4 different inoculant types along with water. And the reason that the treatments were doubled, it was then a duplicate, was because I was collecting tissue at a time course at 12 and 60 hours after inoculation for later RNA sequencing. And I wanted to sample destructively, right? I didn't want to be harvesting tissue from a tomato, waiting a couple days, and then pulling it again. I didn't want to, you know, change expression that way. So everything was done in duplicate, so I could do 1 plot once and 1 plot another time. And then all the plants were assessed for disease at 144 hours. That's when they were really sporulating disease was noticeable and reasonable to assess. This is just a layout of one of my field trials. You can see the 3 blocks with the randomized treatments within it. So like I said, there was 2 of those. And they were done a week apart, and they were planted independently. They were inoculated independently with the wrong run. It was really 2 different experiments. The lab trials, of which I had 3, were set up in a very similar way. I inoculated detached leaflets in moist chambers, again with the sprayer. It had the same treatments. It was again done in duplicate, so I opened a plate, harvested, and I didn't have to open that plate again because the changes in humidity can change things again. And just like the field plants, they were assessed at 144 hours after inoculation for disease. So what did I see? So for the field trials, at 72 hours after inoculation, I actually went out and pulled the plants. I dug them up, and I put them in big plastic trash bags with wet paper towels to keep them humid. I would have loved to have left the plants in the field and let them sporulate naturally, but this is a working farm. It was early in the season, and I didn't want to be this huge source of inoculum for everybody doing trials around. And I mean, I myself had a vested interest in it. After the first run, I had my second experiment to be done the next week, right next door. So if these sporulated in the field, I was going to cause problems. So I bagged them and brought them into the lab where they were kept at room temperature until 144 hours, where they were assessed. to just let them sporulate out. And so I took cuttings from the plants. They were quite large plants. They took terrible pictures this whole. So I just sort of lopped the top off to take these pictures for you guys. And so here we have a plant that was inoculated with inoculum from Newark Valley that was grown up on plates, and a plant that was inoculated with Newark Valley that was grown up on leaves. We can actually, could we turn these lights off? I don't know if the slightest idea how to do it there. Excellent, thank you very much. There we go, so we can see much better. So inoculated from plates and inoculated from leaves. And they're just a sample of the plant. And so you can't see it with great detail. But the disease in the Newark Valley plate is significantly less than on the leaf. This was rated around 20%, whereas this was much, much higher. The differences are even more exaggerated, though, when we look at the older isolate, 140112. So this plant was inoculated with inoculum derived from the plate, and you can almost not see the disease. There's a discrete lesion here, and there's a few little lesions within the leaves. But as compared to its leaflet-derived cousin, I mean, the disease on this is pretty significant. It's really, the plants were really, really heavily diseased. The lab trials were assessed very much the same way. I assessed them in 144 hours, and I took pictures, though actually the pictures you guys are about to see are photos taken at 168 hours. The first run of photos I took, for actually the first 2 runs, were just like this. The top of the moist chamber was on top of them. And I realized actually, luckily, after the second lab trial, when I looked at them on my computer, you couldn't see anything. I saw it when I did the assessments because I opened them up. The pictures were no good. So I quickly noticed that I had a problem. Luckily, I saved all the plates, and so the next day I just opened them up and took much better pictures. So the disease you'll see in the pictures is a little bit higher than they were rated at. So in the same order, we've got our Newark Valley plate-derived and a Newark Valley leaf-derived. And you can see the Newark Valley plate-derived are quite a bit healthier than the Newark Valley leaf. And if you can't see because the resolution isn't so great, if we zoom in, the Newark Valley plate-derived is quite a bit of healthy tissue still, and the tissue is still rigid. And though it is sporulating, it's still quite intact. Whereas the leaflet-derived is completely water-soaked. You couldn't pick these leaves up. They would just disintegrate. And it's absolutely covered in sporulation. Are you gonna? I don't know. I don't know how to do it. Oh, there we go. Excellent. So yeah, so if you can see, this tissue is just completely covered in sporulation, whereas this is plenty of healthy tissue. But the difference is, again, even more magnified when we look at the older isolate, 140112. And actually, if you'll notice, the overall disease is a bit magnified in these lab experiments. These moist chambers are ideal conditions for sporulation, so the differences tend to get a bit exaggerated. But so if we zoom in again on the 140112 isolates, the plate-derived inoculum, it's essentially healthy tissue. In this plate, there was one discrete lesion that you can barely make out of the projector here, right there, as compared to the leaflet-derived at the same concentration. I mean, it's completely water-soaked, completely covered in sporulation. So there was a really, really notable difference in disease. And so if I just accumulate the averages across my trials of these disease ratings, so for the field trials, obviously we had no disease in the water controls. And if you look at within the Newark Valley, the leaflet-derived inoculum was on average 40 and 30% respectively for the 2 trials, as compared to 15 and 10 for its plate-derived version. And the same pattern held true with 140112, the older isolate. The leaf-derived inoculum was in the 50s for disease, and the plate-derived inoculum was down in single digits as far as assessed disease. It was a really notable difference. And if you're wondering about these averages, about how consistent it was across the reps, so these are all the assessments, and you can see that this was within trial 2. You can see the numbers are quite the same all across. It was really, really consistent. The lab trials was exactly the same way, although a bit more exaggerated because of the nature of these lab inoculations. The water controls were clean. The Newark Valley leaf resulted in about 80% disease at 144 hours, as compared to its plate-derived inoculum, which only resulted in about 30%. And then again, more exaggerated with the 112, the leaflet-derived was around 90% disease as compared to single digits. So it was a really significant difference. And all these assessments and inoculations, purposely I did them blind. I had a number system that was just randomized. I didn't know what I was spraying on what, I didn't know what I was assessing. And so I have a big notebook page all filled with these numbers that I wrote down directly. And then after I collected all the data, I went through with highlighters, color-coded, and set it up. And just this pattern showed up, and I'm sure Bill has an email somewhere in his inbox of me just being ecstatic at 2 in the morning one day that it just fallen together so beautifully. It was really exciting. So after I had these very promising results from the phenotypic analysis, I wanted to move on to the gene expression, to actually looking what was behind this. So I've discussed the tissue collection. I sampled in the field, and immediately froze everything at all the time points. And that was brought back to the lab for total RNA extraction. So for the infected tissue, I had tomato host gene expression with the Phytophthora gene expression mixed in. And from those, I prepared my libraries for for RNA sequencing, to do the RNA sequencing, and then I also did some real time to just try and get a handle on what was going on in these samples. For an overview of all the samples that I collected and of all the samples that I prepared for sequencing, this is the first field trial. You can see the 3 blocks, 2 time points, and the treatments within them. 112 PNL being 112 plate derived and 112 leaflet derived, and the same for New York Valley, plate and leaflet derived. So there were 2 field trials with 3 blocks each, although I lost one of the blocks to disease. It was really close to an adjoining field that had disease. I didn't really see disease in it, but I culled it just to be on the safe side. And I didn't want to have any chance of unexpected contamination. I have my 3 lab trials, which were all done independently with the same setup. And then these samples have become really important as the experiment has moved on. These are just the inoculum, so just sporangia collected from 14012 in plate or 14012 in leaf, and the same for the New York Valley in plate and leaf. It's not infected anything. It's not been spread anything. It's just the concentrated sporangia collected from these different inoculum sources that I was working on. So again, the RNA was there for RNA sequencing and for the real time. Because I have so many samples, these cDNA libraries became a really, really non-trivial task, right? Of all the RNA that you extract from a sample, quite a bit of it, a majority of it is going to be ribosomal RNA, which I'm not interested in for my differential expression. And so of that fraction of that that's going to be useful messenger RNA, only a small fraction of that in my tissue samples is going to be Phytophthora RNA, right? The vast majority, by weight, is going to be tomato. And so really getting good cDNA libraries was important. And I actually, the solutions that I ended up using, I actually just think is really interesting, so I'll tell you about it. So it exploited the fact that messenger RNA has a polyadenylated tail. And so you can use ferrite beads that have a probe that associates with that tail. And then once your mRNA is associated with these beads, you can just apply a magnet and wash everything else away. And so all you're left with is your mRNA that you're interested in working with. It's very efficient at getting rid of all that ribosomal RNA. And it gave me the best chance of actually getting my small fraction that is Phytophthora RNA. And then it's all nice and bound up, so you can chop it up with starters and stoppers into small fragments usable for sequencing, and produce your double-stranded libraries, and then associate barcodes, because I did a fair amount of multiplexing. I have a little over 100 samples, so multiplexing became really important for me to be able to run more than one sample in a lane, so I could actually look at all of my samples. Now, unfortunately, because of some holdups with the sequencing, my RNA libraries are currently waiting to be processed at Weill Cornell down in New York City. So I haven't been able to do my RNA sequencing analysis for you guys yet. But I certainly couldn't wait that long to be able to look into what was going on. So I set out to do a whole bunch of real-time PCR. I just wanted to get some sort of an insight into what was going on with my samples. I made RNA from the total RNA extracted from my field samples. There was no ribosomal exclusion or anything. It was just all the RNA that I had. It was made in cDNA so I could do real-time on it. For tomato, I looked at a handful of genes. I had elongation factor mostly as a normalizer, as a reference. It's a housekeeping gene. It's good. It's a good way to normalize my read back to. And then I had Chi3 and STH2. Chi3 is just a chitinase, and STH2 is a generic pathogenesis-related gene in tomato. I'm not expecting them to have a direct relationship to the Phytophthora infection, but they can give an overview of how the plant is responding to an infection. I just wanted a little window into how the tomato was behaving. For Phytophthora infestans, I normalized everything back to ITS, which is just a transcript spacer. It's a good normalizer because it's not really differentially regulated. I looked at an ATP binding gene, a putative RXLR that we've looked at a number of studies. It's just a really upregulated effector at early stages of disease. And INF1, which is an elicitin, which is adversely really upregulated at late levels of disease. I was hoping it would give me a good insight into what was going on. So this is just an example of one of the real-time runs that I did. This was on 60-hour leaf tissue that had been inoculated with 140112 from the plate, leaflet, and the water control. This was a lab experiment, so there was a really big difference between the two. And when we look at the tomato genes, there's really no difference. The tomatoes aren't acting differently. At 60 hours when this was harvested, we have to remember that we're more than 60 hours away from my assessment. So we're quite a ways off from actual disease. The tissue is green and healthy. It doesn't really look like it's diseased. And I wasn't actually super surprised by this, that they're not really acting differently, especially the plate-derived one, because that one, by 144 hours, it was barely diseased anyway. If we looked at the Phytophthora expression, though, at first glance, it looks like there's a difference. I mean, there are differences in expression. But I think that this can actually be explained in a different way. So while the tomatoes were indistinguishable, the Phytophthora infestans expression is just sort of relative to its stage in its disease progress, not to its time point, but how it's progressing in disease. And by that I mean if I aligned the overall disease that I ended up with this exact sample at 144 hours, right? The leaflet-derived one, we ended up at 90% disease, as compared to only 1% with the plate-derived one. So they're at very different stages of their life. And to exaggerate that problem, right, with the RNA extraction and with the real-time PCR, there's a detection threshold, right? There has to be at least a certain amount of material there for you to be able to see it. And for the small fraction of the RNA that Phytophthora is going to be, it may very well be that at this stage of disease, there's just not enough Phytophthora in the lower disease for me to really be able to see much. So to put that more graphically, our leaflet-derived plant at 60 hours was pretty close to disease. 60 hours after the sampling time, it was sporulating pretty heavily. Whereas the plant inoculated from plates, 60 hours afterwards was still a green, healthy plant. At 144 hours, it only had 1% disease. But if I let these plates go, and actually in most of the experiments I did, I let them just keep running. The 90% disease ones, they didn't get any worse. They couldn't get any worse. But by a week later, the plates that had only 1% disease, 5% disease at 144 hours had caught up. The disease had spread and they were now totally infected. So I've just got this little window into the overall disease, and I'm just seeing expression relative to that. And so while it was maybe a little bit discouraging, at least for the real-time PCR, and I had to wait to really get some insight into this until I could do my RNA sequencing, what it did do is redirect my focus. I had these sporangial isolates of just the sporangia, and I really thought that if I was going to have any chance of seeing actual differential expression, different things going on, it was going to be in those sporangia. Because, as we discussed with our arachidonic acid experiment, these sporangia are different somehow. They behave differently. And this is just an older experiment from our lab back in 1999. And it wasn't looking at inoculum sources. It was looking at sporangial survival in the face of solar radiation, right? So how well they survived in the sun. And if the experiment was done with sporangia that came from pure culture, the results were all over the place. They didn't survive very well, and it wasn't very reliable. But Eduardo, who was doing the study, noticed if he was using leaflet-derived sporangia, they survived much better. He got really reliable results. And so without even looking into it, he had really noticed that these sporangia, just on their own, were behaving quite differently. Somehow they're surviving differently, so they're expressing things differently. So I refocused on the sporangia, and I did some more real-time PCR. So I checked those genes that I was talking about, and these are just their expression levels normalized to the ITS, which I was talking about before. And the ATP binding cassette is really, in Phytophthora, it's really reflective of just cellular activity. And the Phytophthora is really up and doing things and really going. But you see more expression when it's latent. And the expression was not different between the two of them. The sporangia are expressing it at the same levels. They're They're just sporangia, right? They're waiting for an effect. But when I look at my RXLR, my effector, we saw a fair difference in expression. The leaflet-derived sporangia had a twofold expression. They're expressing it twice as much as the plate-derived sporangia. And then when I investigated INF1, that elicitin that's associated with late-stage disease, the difference was just astronomical. So if we look at ratios here, the plate-derived sporangia are expressing this elicitin at about 100 times more than their leaf-derived cousins. And I actually ran this a couple times, on a couple different RNA samples, because I wanted to make sure that that's what it was. But these ratios hold true. And the INF1 is really interesting in this. So INF1 is like the primary secreted elicitin in Phytophthora infestans. It's talked about quite a lot. It's associated with late-stage disease, inducing hypersensitive response. It's not known to be the cause of resistance to Phytophthora infestans in tomatoes or potatoes, but it is likely the reason behind some non-host resistance in some tobaccos and some other plants like that. It induces this HR. But in Phytophthora infestans, what it is associated with is really high expression at late stages of infection when the tissue is necrotic and it's sporulating heavily. And it's also associated at really high levels with culture growth. If you look at Phytophthora growing in pure culture, because it's not growing on a plant, it doesn't have to worry about eliciting responses. The pattern is that it's really highly expressing this when it's in culture. It doesn't totally stop expressing it when it's infecting. At early stages of infection, if you look at biotrophic early Phytophthora growth, it's still expressing this gene, but it's at much, much more downregulated levels, levels that were more in line with my leaflet-derived sporangia. But I was a little concerned because they did say that in their experiment, this was back in 1996, that they didn't really see it in sporangia. I was worried that maybe it was some sort of an artifact that I was seeing it. So I made sure that when I collected my sporangia for RNA extraction, I did it exactly like they did in their paper. And I washed it from the plates or from the leaves, just to make sure that I wasn't picking up more mycelia than they were, something like that. And I did my RNA extractions from those to run the real-time PCR. And with my real-time PCR, I checked with melting curves to make sure that the primers that I had designed for this, it was getting exactly what it was. And I really, really was. If I had to try to come up with some explanation, it may simply be that in 1996, with the RNA extraction protocols that we had, and they were using northern blots to associate the genes, it might have just been slightly below the detectable thresholds. I'm not seeing tons of transcripts of this gene. I'm just seeing a differential expression between the two of them. And so the reason this was exciting, and the reason that, again, I fired Bill off a frantic email at 2 in the morning with writing that was hard to understand because I was so excited, is because if we look at the overall expression, the leaflet-derived sporangia have their RXLRs upregulated and the INF1 downregulated at levels that we associate with early-stage Phytophthora infection. It looks very much like when we look at gene expression in Phytophthora infecting well, being a good biotroph. But the plate-derived sporangia, their expression looks an awful lot like late-stage necrotic Phytophthora or Phytophthora kind of lazily growing on a plate. And I'm not making the argument that the upregulated elicitin and the downregulated RXLR are resulting in some sort of resistance or something. It's just that in order for the plate-derived sporangia to infect and really start causing this early biotrophic disease that Phytophthora is so good at, it has to really significantly change its gene expression. It's got to downregulate that stuff. It's got to upregulate the effectors. Whereas the leaflet-derived sporangia are already expressing just like we would expect them in early-stage disease. So I started building the hypothesis that the gene expression in these tissue-derived sporangia, the sporangia we wash from leaflets, is already at what it needs to be a good early pathogen. It's already set and ready to go. So as soon as it is applied, it can start growing. In about 144 hours, we have this overwhelming disease, right near 100% with the lab experiments. Whereas the culture-derived sporangia, they're just not there yet. They just need to take some time to change the regulation before they can really effectively infect. And that may result in that shift in the disease that I had with those arrows, that skews the window that we're looking at. It may just set them back a week or so, which is actually sort of like what we saw in the lab, the plate experiments. So in summary, for the phenotypic analysis, the leaflet-derived inoculum caused a significant amount more disease than the plate-derived inoculum with the same lineage. And this held true across all of the trials in field-type experiments and in lab-type experiments. And it held true for both isolates. It's notable, and it was pretty apparent that there was a difference between the two isolates. And it, again, was the same through all the different trials that I did. And it may have something to do with the time in culture. 1,4,1,2 had been growing in culture for a year before I used it, whereas the Newark Valley was right off of being a pathogen out in the wild. But I can't make too many conclusions about what time in culture might induce. I would need an awful lot more isolates to look at to really make some firm statements on that. And that might be an exciting thing to do in the future, but I couldn't make those assessments with just the few that I had here. The real-time PCR basically told me that with the methods that I have right now, at least with the real-time PCR, I can't see much difference in my tissue samples. I'm going to have to wait for the RNA sequencing to see if I can see anything different. But even then, it may be the fact that in my tissue samples and my time course samples, I'm just seeing the difference in disease stage between the two. What it really focused is that the sporangial RNA sequencing and the sporangial real-time is what's really going to be the crux of the difference between these two. And so for the RNA-seq, as I said a minute ago, I'm still very keen to look at my tissue samples over the time course. And I'm really interested in what's going on, but they may just reflect the disease progression, whereas the sporangial gene expression is really going to be how I'm going to be focusing on the time going forward. They look like they're really quite different, and I'm really excited to see how they're different. And so that's it. I wanted to thank everybody in my lab, all the current members, certainly in the past members who have left, Giovanna and Richard, all of the graduate students, especially the members of the Perry Lab who I've been bothering incessantly for help with the RNA sequencing. They've been very, very helpful. Certainly the folks at the farm in Freeville, the Thompson Research Farm, Steve McKay and the staff there. I've done quite a few field trials with them, and they put in an awful lot of work for us. And the folks down at the greenhouse, and everybody who's helping with this. So thank you very much.
So if you're using 5,000 sporangium per mil and spray, that's maybe 50 per square centimeter or something. I mean, I guess the question is, do you see, if you look at the microscope, do you see different behaviors of those sporangia in initial germination and infection?
So I haven't assessed them microscopically. I haven't looked at that. It would be difficult to see them on the leaf. I don't know. It would be a really interesting thing to look at. And we have done germination experiments with other isolates. We've done it between clonal lineages a couple times to see how quickly they germinate. And that might be a really good thing to do, actually looking forward, even if it was just on culture or something like that. But I haven't looked at it. And so, no, I'm not sure what they're doing immediately when they're on their new tissue like that.
But presumably only a fraction.
Yes. Yeah, certainly not all of them germinate. And the inoculum that I'm spraying, because I let them germinate in the fridge for a while, it's more of a mixed suspension of sporangia and zoospores. There's quite a bit of zoospores swimming around in the water. So the zoospores are applied directly to the leaves, and they very quickly find the leaf. So yeah, I was looking at that, which are actually quite a bit smaller. But yeah, I would expect that only a fraction of them survive, but I'm not really sure what the spores for the ones that I don't see disease for quite an extended time are doing in that time. I'm not 100% sure.
Do you think that the difficulty in taking the sporangia from pure culture and going into plants is related to the same phenomenon there?
Yeah, so it certainly could be. There are a couple of things contributing to that. The simplest, some of them are actually just a mechanical problem. One of the problems is if you were to transfer like an agar cube of a culture of Phytophthora onto a leaflet, it has a tendency to just really grow out of it. It fluffs up. You get a cotton ball of Phytophthora in the agar cube. It preferentially is growing on the media. It doesn't want to go into the leaf. It's difficult to grow into a plant. So when if we make sporangial suspensions and inoculate that way, it's a lot more effective. But it is tricky, and it certainly may have to do quite a bit, with this delay in infection. If you've got a detached leaflet in a moist chamber, And it's sitting, at 18 degrees down in our cold room in that moist chamber, just the leaf. And there's no disease by 6 days, right, or by a little over a week. Then you start running into problems with the cells start apoptosing from the water. They sit in this water agar, and the cells start to explode, or they can get bacterially contaminated, or just start to degrade from not being on a plant for that long. And so one of the challenges with that is just that the tissue gets crowded before we can isolate anything good from it. I suppose if we tried to wake up our cultures by spraying them onto field plants, maybe it would be a bit more reliable. I mean, I reliably got disease off of my culture. It just took a while, but we don't really have that luxury when we're trying to just wake it up in the lab for experiments. But it certainly could contribute, and maybe it'd be part of it, yeah. It would certainly explain why it's such a pain. I don't know if you've got a question?
Yeah, Sean, one issue, two questions. The phenomena you're describing when you first presented some of the quantitative PCR data, where you saw different levels of disease, and you saw perhaps an order of magnitude difference, and you were suggesting that was perhaps an artifact of just not having much disease or pathogen nucleic acids. But was the issue there that you were at the threshold of detection? And the reason I'm wondering is because you seem to have an internal control that you were normalizing it to. And presumably the same question is going to come up when you look at your large-scale sequencing data.
Right. No, the threshold of detection I was talking about, I think is more to do with it. If I was to look at a bigger variety of genes, some of those lower expressed genes that may be more intricately involved, I don't think I would be able to see them at that low level of disease. I think that the phenomenon that I saw there, you're right, it was normalized out to ITS, so that was normalized to the amount of material that was there. So that's not a detection problem. That was just truly representative of the amount of material that I had there. So that was normalized to a Phytophthora gene. So it was just more of an overall quantifier of how much was there. If I had normalized it to a tomato gene, for instance, then we would have gotten some wild things. But it was, yeah, it was internally controlled for it. So it was just reflective. Those genes were being expressed at the same level, the two samples. The quantity that I saw, the difference was just how much was there, I think, if that makes any sense.
And when you get to your large-scale sequencing data, presumably you're going to run into the same issues.
So yeah, I certainly might, and I'm going to have to spend a lot of time working on trying to tweeze out absolute numbers. Right, finding normalizers is going to be really important for the RNA sequencing. It should also be more sensitive than this real-time was too. And actually, in the limited time that I had, I didn't get to spend as much time optimizing real-time as I would have liked to. It has a lot of way to go with the real-time, actually, to be able to really, really rely on it heavily. And it's just been a matter of time. I'm not sure if I answered it very well, but John?
Hey, Sean. So you said that your tissue samples, you believe, are reflective of a delay in disease progression.
Mm-hmm.
So when you look at them, inspect them again later, and you find that they've reached the same level of disease, do you think that the plate-derived inoculum disease is due to the primary inoculum, or is this now secondary inoculum that has grown on those leaves?
That's a really good question. So in field plants, if I had let the field plants go much longer, I suspect that there may have been some problems with secondary inoculum. In the lab experiments, there's no air movement. Sporangia aren't really going to be. As they grew out, they just progressed from, just sort of arose. I don't think that there's any secondary inoculation going on over the short time that I'm looking at with these isolates. Especially, so the lab experiment where I had 1% in 144 hours, there's barely any sporulation. That's not going to be enough to somehow go out, infect, and then in a week later cause a much heavier amount of disease. So I don't think that secondary infection played much of a role in that. I think that it was truly just sort of a delay. It didn't get going very quickly. And I think you had a second part of the question, though. I don't remember what it was.
I think that was it.
That was it? Okay.
Thanks.
Do you think there's a nutrient difference between sporangia growing on leaves compared to sporangia growing on plates?
So like they've been provided with different amounts of nutrients when they're growing, or do they
Kind
have
of
different?
ephemeral nutrients is lost when you make new plates.
I mean, it may be. I mean, the agar that we grow on, potato agar, rye agar, and I used a rye and pea agar mixture. I mean, that's not, right, it's not tomato leaves. So yeah, there certainly could be a difference in nutrients. There could be some, have some effect on that. I'm not sure. I haven't looked into the available nutrients for these though. Anybody else? Nope.
So before we thank Sean again, I'd like to ask one question. How long does it take to prepare 100 samples from leaf tissue to send to Cornell?
I can do 8 at a time with the kit that I was using, and that protocol from straight RNA to a finished DNA library takes about 5.5 hours. And so 100 samples across 5.5 hours, doing it every day.
How long does it take to get the RNA?
About 3 hours, 3.5 hours from sample. It's a long process.
Oh, I was shorter than I thought. Why didn't you do it faster?
There's a lot of checking and rechecking in between.
So, thank you, Sean, and see you next time.
This has been a production of Cornell University, on the web at cornell.edu.